I'm so bored. It's really hot out today. I'm just gonna go back to TikTok.
Come on, there's more to life than just TikTok.
Like what exactly?
I'll show you something that was cool way before TikTok, but we'll have to go outside for it.
I don't know about all that.
Come on, you'll be energized in no time. Let me set the scene. It's 2014, and Grumpy Cat is prowling the internet. The world is learning how to let it go because Frozen has just come out, and another viral sensation is taking the internet by storm. The Ice Bucket Challenge.
No, we're not doing that.
It's important to look through history and consider the important events of the past. ALS is a rare but severe neuromuscular disease. It's what Stephen Hawking had. The world raised awareness by doing the Ice Bucket Challenge. All we need is a little ice.
That is cold. No.
Cold is just a sensation, that's all I learned.
No, cold is a complex physiological process that involves sensory neurons that innervate the spinal cord and higher regions of your brain. You can't just say that.
Where did you learn all that?
It was at this sensory physiology club.
That sounds lame, but I still want to learn. But first...
No.
Hello, dear friends! Welcome to our game show.
Cut!
Hello, dear friends! Welcome to our game show. Oh. Hello, dear friends! Welcome to our game show Ice Buckets versus Scientists. Today, we will help a confused group of scientists to discover how people feel cold. Together, we will guide them through using the scientific method to obtain new knowledge about our universe. At the same time, we will explore our beautiful lab, our equipment, and we will learn how some of the common biological methods work. And now, let's meet our fabulous cast!
Hello, I am Scientist 1. In lab, I like to multitask.
Hi, I'm Scientist 2, and I eat pieces of science like you for breakfast.
Our journey today will take us through several stages of the scientific investigation. And if you successfully complete each stage, you will get to see how our scientists celebrate in a very special way. Believe me, it's worth it. So pay attention, think, and participate. Every story has a beginning, and every scientific story begins with a good question that people are curious about. Scientific questions usually take the general form of how does this thing work? Or alternatively, what is the mechanism? You probably heard the expression standing on the shoulders of giants. And it is especially relevant in science. Throughout the ages, people have already learned a lot about the world. And therefore, the first place to try to answer your scientific question is to examine the literature and see what others before you have already learned and reported.
Hey, what happened to you?
Oh my god. Oh my god. I was up all night reading literature about how we sense cold.
And did you learn anything?
Actually, yes. We already know a lot of stuff. I found some articles and also an online video by this guy named Viktor or something.
Basically, what he said was that cooling activates nerve endings in the skin, which belong to sensory neurons. These neurons then transmit the cold signal to the spinal cord. The signal is transmitted across a synapse to a spinal cord neuron, which projects to the brain, where the perception of cold is produced.
Okay, but we need to dig deeper. Did this guy Victor say how the neurons get activated by the cold in the first place? Like, how do neurons feel cold?
He didn't say, but I read that there are proteins called receptors. They sit in the plasma membrane, or the outer layer of the neuron, and are activated by different things like temperature or chemicals. When enough of the receptors are activated by a stimulus, the neuron fires and a signal is sent to the brain.
So to feel cold, we probably need to have receptors present in the sensory neurons that are activated by cold, right? I wonder which receptor is responsible for being sensitive to cold. Do you have any candidates?
I found some papers, but I really can't read anymore. I need some help.
I hypothesize that the receptor M8 is responsible for cold sensation. If my hypothesis is true, and the M8 receptor is responsible for cold sensation, then receptor M8 must be 1. Sufficient, and 2. Necessary for cold sensation. By sufficient, I mean that if we put the receptor in any cell, and that cell becomes sensitive to cold, the receptor is sufficient for sensing cold. By necessary, I mean that the receptor has to be there for cold sensation to work. If we get rid of the receptor and cold can't be sensed, then it is responsible and necessary for feeling cold. Scientists call getting rid of a gene knocking out a gene. If we knock out a receptor and cold can still be sensed, this means that the receptor is not necessary for cold sensation.
It's recording, right?
Yeah.
Should I make it nice and slow for you? Are you recording?
Oh, whoa, right on the head. Oh.
Get it on in there. Revenge. Revenge tastes sweet.
Each neuron has genetic material made up of genes, or instructions, which make different proteins, including receptors. Genetic material is stored in a cell in the form of DNA. We can think of DNA as a recipe book, and the many genes or instructions which make each protein as a recipe. To transfer the cold receptor from a sensory neuron to another cell, we need to first learn the cold receptor recipe, or gene, from the sensory neuron. Since we don't know exactly which receptor is responsible for sensing cold, we will learn the recipes for the M8, A1, and C5 receptors. We can then give these recipes to a different cell, who is a bad cook, who doesn't know very many recipes. With the new recipes, the bad cook cell will learn to make the receptors for itself. At the end of the day, we will have 3 different cells that have learned to make one receptor each, M8, A1, or C5.
Since cells can't talk, we have to use a different method to determine whether they feel cold. We will use an experimental technique called calcium imaging. Yesterday, Viktor told us that when a neuron is activated, there is an electrical action potential. This causes calcium to rush into the cell. We give the bad cook cells that now express a new receptor a special dye that glows green when calcium enters the cell. We can tell when a cell is activated because it will glow green. Therefore, we can expose the cells to cold. If they glow green, it means they are sensitive to cold, and therefore that the receptor is sufficient to sense cold.
Hello everyone, and welcome to our lab. Today we'll be testing whether the bad cook cells that we've taught to express the candidate receptors A1, M8, and C5 are activated by the cold. To do this, we'll be using calcium imaging. So let's take a look at the equipment. Follow me. In this 12-well plate, I have small glass discs upon which the bad cook cells are attached. These cells now contain only M8, A1, or C5 receptors. We have also treated these cells with a dye that causes them to glow when they are activated. In this corner, we have the calcium rig, where we can perform calcium imaging. Let's take a closer look at each of the parts. The first thing that we need is a microscope to allow us to look at the cells. This is because the cells are too small to see by eye. Attached to the microscope, we have a camera, and this camera allows us to look at the cells on a big computer monitor. Here are our cells here. This also allows us to record the experiment so we can perform analysis later. The second thing we need is a place to put the cells while we apply cold. We put them in this small area here, which we call a bath. Bathing the cells in a cool solution is a convenient way to cool them. To supply the cold liquid, we use this perfusion system. This system is made up of a series of syringes here that we can fill with a liquid. The liquid travels through these tubes to supply our bath with liquid. This device here is a Peltier, or a fancy machine that allows us to cool the liquid to the temperature of our choosing. To perform the experiment, we take the cover slip that have cells attached and place this cover slip into the bath. We then supply the cells on the cover slip with room temperature solution through the perfusion system. This is the control condition during which we expect cells to not be activated. We then supply the cells on the cover slip with cold solution through the perfusion system and record their activity. If the cells are activated by the cold solution, this means that the receptors that the cell contains are likely sufficient to feel cold. Now, let's go perform some experiments and analyze the results together.
We will knock out our candidate cold receptors M8, A1, and C5 in mice and determine whether they can still sense cold. These are specially generated mice that are normal in every way, except they lack one specific protein. What happens if our animal is a knockout for a gene responsible for feeling cold? This is what we are going to find out today, using 3 animals with 3 different knockout genes, C5, M8, and A1. If one of those genes is responsible for feeling cold, we will be able to find out using the 2-plate preference test. Good morning, students. My name is Marco, and this little fella is 3156. And today we are going to show you the 2-plate preference test. This apparatus is made by 2 plates, and we are able to control the temperature of each plate. For example, as you can see from the thermographic image, this plate is colder, 60 degrees, while this one is warmer, it's 80. Now, what happens when the animal enters the apparatus? Well, first he will explore, and then he will decide which plate is more comfortable for him. Let's see what happens. One plate is cold, and is cold and is 60 degrees. For him, is like chilly, like for you when you enter the frozen food area of the supermarket. You probably will grab whatever you need and then run out. For the mouse, should be the same. Hi again, I'm Marco, and I'm going to teach you how to score the 2-plate preference test. To do that, you need 2 things, a stopwatch app and fast fingers. In this experiment, we have 2 plates. One is warm and cozy, while the other is cold and chilly. I will put the animal in the middle of the 2 plates, and you will start moving freely around. What you have to do is to take the time when the animal enters into a specific plate. For example, let's say plate 1. So, when the animal is in plate 1, you start the timer. When it crosses the middle line and goes to plate 2, you stop the timer. You keep your eyes on the animal, and when it crosses back, you start the timer again. Rinse and repeat for the whole duration of the experiment, that is 3 minutes. So, to summarize, animal in plate 1, you got your timer running. Animal moves to plate 2, you stop your timer. In the end of the experiment, your stopwatch will tell you how much time the animal has spent in plate 1. And from that you can calculate how much time the animal has spent on the other plate, plate 2. Now, knowing that one plate is warm and the other is cold, and knowing that probably the animal prefers the warm one, can you tell me which plate is what? And if one animal is not feeling the difference, could you tell me what animal is not feeling cold or warm? It's up to you. So the question is, like the mice, can I feel cold, or I will be totally indifferent? Let's try.
So that's what I learned at Sensory Physiology Club.
That sounds pretty cool, but not as cool as this!